a goal is rather to reach to the end point during a race. Both are goal-directed locomotions in a sense that the image on the left also has a goal to reach to safety. So the threat and safety are two parameters which are present everywhere in the environment. And the organism, all the organisms have the ability to sense these aversive stimuli and in order to execute escape and avoidance behavior. And also they have sensory abilities to sense appetitive stimuli and approach toward these appetitive stimuli in order to execute various adaptive functions. And this is very well evolutionary conserved, and even can be seen in a single cell organism such as amoeba, for example. They can sense charge, they can sense, they have chemotaxis. So, so they can movement toward and away from different sensory stimuli. It's also, it can, I think for college students, I mean, you must be familiar with earthworm responding to aversive stimuli in a tea maze, for example. And those responses can be conditioned such that the animal does form an aversive and avoidance behavior. And it continues throughout the species spectrum all the way to humans. So why do animals execute such movements, complex movements? The reason behind that is in the environment, the animals use this threat and safety signals via sensory mechanisms and execute complex motor movements to navigate in these environments. And when the threat is imminent and extreme, the survival response is paramount. The animal has to execute a survival response, and it could involve a very primitive reflex response, such as like a knee-jerk reaction, or also if you touch something hot, even before your brain can decide, it doesn't wait for that. It actually go through a reflex arc, and then you just move your hands away. So it could be as primitive as a reflex action, and it could be as complex as a coordinated escape, which involves running and jumping and avoiding obstacles on the path and reaching to safety. So, this is a very wide spectrum. This involves very wide spectrum complex movements. And the other hallmark about this acute presentation of threat is that the animals do not keep executing these complex movements. And once the threat is, the imminence of the threat is gone, the animal will stop freeze, flight, or fight behaviors, and they will tend to go back to the environment and start foraging and feeding in the absence of any imminent threat. However, we all know that in the environment, there is always a presence of predatory threats to the animals, but they do not keep reacting on just because the threats are present. They just react when the threats are imminent. And they have to do the survival response. So I'll show you this video where this is a video of a wild mouse. It's eating in the wild, and pay attention when a visual stimulus comes in via visual or auditory stimulus. The animal just freezes for a moment and then execute a very rapid escape. But it doesn't stay there. It doesn't keep doing the escape. It reaches to the safety, but when the threat has been resolved, it does come back to its feeding ground again and resumes its behavior. So a couple of hallmarks of this complex behavior, that on the visual stimuli, the mouse stopped or froze for a moment on the presentation of visual or auditory stimulus, And this led to termination of all ongoing behaviors, which includes feeding in this case, for example, because the survival is paramount. So the animal has to stop all the movements and only select complex adaptable movements, which will be helpful to execute a successful escape. And the other hallmark behind this is the animal does come back really quickly to see if the threat has gone, and if there is no further threat, the animals will keep feeding. And this is how things work in a very primitive level in environment. But this is true even in every organism in the species. So why we need to study these underlying neural circuits behind these complex adaptable movements in response to threatful stimuli? The answer is, if you carefully look, many of the brain disorders involve motor and cognitive dysfunctions, which are hallmark of many brain disorders like Parkinson's disease, ADHD, panic disorder, phobias, anxiety disorders. So you will often see the cognitive dysfunctions accompanied in neurodegenerative diseases are also accompanied by some lack of execution of some complex adaptable movements. And also the fact that many medications aimed to improve either motor function in brain disorders or aimed at improving cognitive and emotional functions often result in a side effect which affects either motor or cognitive or emotional functions, such as, for example, some medications to treat tremor, for example, in Parkinson's disease, the side effect may be increased dysfunction in emotional behavior. So we really need to understand what's going behind this neural circuit, which regulate two distinct type of locomotor behaviors, one with a positive effect and one with a negative effect, to discover new targets to treat Parkinson's disease. So this is a very basic diagram. It depicts the spinal cord. Can you see can you see the arrow on the screen when I point? Yeah. So I mean this. So this is a portion of spinal cord which has been shown to execute complex movements such as walking, and it does house a central pattern generator. So the central pattern generator is a nomenclature of neurons present in the nervous system, which can oscillate in a rhythmic fashion without any significant input, for example. So there are many examples of rhythmic motor movements in our body which are governed without any voluntary action. For example, breathing. It does involve a very complex rhythmic movement of motor neurons to keep us breathing. And the CPG, or the central pattern generators, they reside in the lower brainstem. Chewing, for example, is also an example of complex motor movements. Swallowing, eye movements. There are many. Even the heart uses pacemaker function. It's a rhythmic movement. So the central pattern generator, the term refers to the neurons which are capable of generating rhythmic oscillations. And then we know that the lumbar spinal cord is the part where rhythmic these CPGs, or central pattern generator, reside for doing walking and running, for example. Indeed, in the brain, for example, the cortex controls fine motor skills such as speech, dexterity, hand-finger control. Basal ganglia is important in doing providing valence, for example, in complex behaviors such as eating, drinking, and also there are brainstem participants for executing expression of emotions. So So I mean, if this image on the left actually showing a decapitated chicken, and you probably, some of you can recall, I mean, this is a very widespread phenomena. Even when decapitated, animals can still execute complex locomotor movements. Farmers probably were the first scientists to discover that when they decapitated the chicken can still run around a fair bit. And there is another example of such preparation which is done in our lab by Simon Sharples. So this is a decapitated mouse, and on the addition of a neuropeptide dopamine, you see the animal starts to make complex stepping movements resembling to the walking signature. This is also true in case of, for example, when this is another preparation where this animal has been restrained and it has been decerebrated. Much of the forebrain has been gone, and leaving only brainstem behind. The animal can walk spontaneously, and on application of intrathecal dopamine, the animal actually responds vigorously. It raises its posture further and increases the speed of spontaneous locomotion. So the idea behind all these experiments and observation is that spinal cord indeed has capability to execute the complex locomotion, but and it can function in isolation, but it really does in complex environment. Can it really function without the brain? The answer is probably no, because we all need brain for a reason, is to make these complex adaptable movements, just not walking and running. For example, this image show that both predator and prey, they have to execute these complex locomotor movements in order to reach their goal, which could be different on both ends, for example. So what we have done recently in last 4 or 5 years, we have started to study these supraspinal, which means the center or brain centers which are above spinal cord and could communicate with spinal cord to execute these complex locomotor movements. So very at a basic level, very high level, the MRF stands for medullary reticular formation in the brainstem. There are many participants and neural circuits in the brainstem, the lower brainstem, the caudal brainstem, which functions like a switch on and off. If you activate these neurons, and this has been seen classically, I mean, as early as early 19th century, that when you electrically stimulate cats or rats, for example, in this brainstem area, the animals execute complex locomotion movements. So these are like switch on and off button for executing locomotion. If you go a bit rostral in the brain, the upstream regions, this is called mesencephalic locomotor region. This also has been shown to electrically respond with electrical stimulation, and it also produces locomotion. And if you further go in the hypothalamic region, in rostral in the brain, there is also another region called diencephalon or subthalamic region or diencephalic region, which has been shown to respond to electrical stimulation to produce complex locomotor movements. So I will come to this later. We have recently compiled all this information regarding this all neuronal circuits, but the key question is where we start studying these complex neural circuits. So I'll just start with subthalamic diencephalic locomotor region, which I showed, which constitute part of lateral hypothalamus, part of posterior hypothalamus, and a region called zona incerta. So the zona incerta, the literal meaning of zona incerta is zone of uncertainty, and it has been named as such because the functions of these regions are not known. So the incerta means uncertainty, and zona means zone. So it's a zone of uncertainty. However, the very first observation, which was done in 1940 by Waller, He stimulated this brain area with electrical stimulation, and he found that cats can walk when electrically stimulated in this area. This has been repeated many times. One of the classical hallmark, it was shown, it seemed like the animals were making a directed locomotion response, which could be implicated in goal-directed behavior. However, there hasn't been any proof that this area does evoke a goal-directed locomotion, and if it does, which cell types or which circuits are responsible for this. So we'll get to that in a moment. So as I shown you, this region in red is called zona incerta, or zone of uncertainty in the brain, and it has very extensive output connecting to various brain regions and widespread connectivity. But we don't know exactly like what type of cells it has, which neurotransmitters they express, which neuropeptides they express, what kind of, like how much heterogeneity in terms of cellular phenotypes we have in this region. Classically, it has been shown it does have glutamate neurons, it has GABAergic neurons, it has neurons producing dopamine, some somatostatin, even MCH, but we don't know much about the circuits of these individual cell types and how these are related to classical brainstem switch on regions such as MLR, MRF, as I said, and also spinal cord. What are their targets, and what are these functional relevance of these microcircuits? So we have started to take these circuits one by one. I mean, it's not possible to study all circuits at once. So we decided, okay, let's focus on the relevant brain regions here, which are in the brain stem and could be acting as a switch on and switch off functions. So just to introduce, I mean, this is in terms of how neuroanatomical tracing is done in animals, it's generally done using a very simple concept. You must be familiar and you must have read in your higher schools in college. There are molecular motors in the cells, for example. And as you know, the neurons do have a cell body, and they do have an axon, and they have a presynaptic terminal. So there is a molecular machinery composed of molecular motors, which can take like a cargo, like a truck, which can take the stuff from the cell body all the way to presynaptic terminal. It's a one-way highway, and which is called anterograde because these key chemicals or vesicles are taken away from the cell body to the presynaptic terminals. Whereas there is another highway bringing vehicles or chemicals or messengers back from presynaptic terminals all the way back to the cell body, as you see. So if you inject a tracer in the cell body, it will travel to all the presynaptic terminals, and you can identify the possible postsynaptic counterparts in this fashion. Whereas you inject a tracer in a neuron, it would all, which is retrograde and which hijacks the cargo which is going upstream. Those tracers are called retrograde tracer. They specifically hijack a cargo which is going upstream in one direction to the cell body, and then you will be able to label the cell body. So this is how basically neural circuit tracing has been done in past using chemicals. Such chemicals like horseradish peroxidase, fluorogold, or even bioteins. There is a whole class of anterogrades tracing chemicals and also retrograde tracing chemicals. So we utilized these. First of all, we just said, okay, let's do a very simple, basic chemical tracing employed in neuroanatomical circuits, and we will study the circuits which are going away from zona incerta all the way to cuneiform and pedunculopontine and maybe the lower brain stem and possibly to spinal cord. So in order to do that, we chose the region called cuneiform nucleus in the MLR region, the mesencephalic locomotor region, which is in the midbrain. And when you inject a chemical tracer known as fluorogold here, which is shown here in the red, So it does travel all the way back to all the neurons which are sending projections to this region. So what we see, a very discrete region in the hypothalamus or in the zona incerta was labeled following a retrograde deposition of fluorogold in the cuneiform in the midbrain. So we saw a large number of cell bodies in the zona incerta. So the zona incerta was indeed communicating to the midbrain switch on, switch off circuit for locomotion. But the question is, what type of cell these are? All these are same cells producing similar neurotransmitters, all they connected similarly. As you know, like if you neurons do connect to another neurons using synapses. So these all neurons possibly are connecting to various number of neurons in various brain regions and all the way to spinal cord maybe, making a connectome. And this has been revolutionized recently. I will come to that later with the introduction of new techniques in the neuroscience, and which are possible to execute in rodents, and which have brought us unprecedented amount of data, which has led to the need of using artificial intelligence and new techniques using data analysis, supercomputers. So we have been able to generate in last 5-10 years tremendous amount of data, how these brains, how many circuits are there, and it's amazing. Like for more than 1 billion neurons, the possible number of computation and probabilities to connect with each other are tremendous. So we'll go to that discussion later. So we similarly did this retrograde tracing from cuneiform region in the midbrain, which is part of mesencephalic locomotor region. We also did similar studies from the other counterpart of the MLR called peduncle pontine nucleus, and also from the caudal brainstem. We found the zona incerta was indeed communicating to these brain regions as depicted in this sagittal section of the brain. But still the question remains, even you have discovered connection, we still have to discover whether this is a true synaptic connection, or is a monosynaptic connection, or is a disynaptic connection, or it's a polysynaptic connection, you know. So that would mean first order, second order, and third order neurons, as people know, for example, right? So, and what is the functional relevance of having these circuits? And so one of the key observations we made between all those neurons which were labeled and were found to project to these brainstem locomotor switch-on regions, a part of neurons were expressing the enzyme tyrosine hydroxylase. The tyrosine hydroxylase is a rate-limiting enzyme in the synthesis of dopamine in the brain. And that thus putatively, and we have confirmed using the different markers or different enzymes involved in the dopamine synthesis, that some of these cells are indeed dopaminergic. And this is actually a novel or a lesser studied population in the brain, which is also called A13. So the A here means aminergic. And and because these are amines, dopamine is an amine, right? As you know, so there are different types of amines found in brain, and then these were named for their position. So this alphanumeric nomenclature A13 means going from caudal brain, from brainstem to the rostral brain. This is a 13th population, or a 13th island of dopamine cells, which is present there in the brain. And there are around 16 such regions which in the brain, all the way up to A16, which express amines in the brain, monoamines. So, but one of the largely studied such collection of neurons is in the midbrain, known as substantia nigra and ventral tegmental area. So those regions are highly studied, and those regions have and neurodegeneration in those regions, which make part of basal ganglia, is known to produce tremor and Parkinson disease and other neurodegenerative disorders. But we don't know anything or much about what these dopaminergic neurons are doing in the hypothalamic region. So we really need to find their circuits and their function. So as I was studying, like moving away from the ability of chemical tracers to do like a one-by-one examination of circuits, this opportunity in last 10 years has been expanded with the introduction of a very simple discovery or observation that jellyfish or organisms, other organisms, they do have fluorescent proteins. As you can see, it's a very nice picture of jellyfish expressing green fluorescent protein. So ever since the green fluorescent protein was harvested from jellyfish and it was used to trace neural circuits, we saw a tremendous growth in our ability to detect neural circuits. So how it's done, so this the green fluorescent protein, it can be packaged into adeno-associated viruses or another viruses, because it's ironic to discuss about virus during COVID pandemic, but the viruses are also friends of a neuroscientist, because the viruses are known to attack different cell types, including neurons, and we can harvest, and we have been able to harvest this ability to attack particular using particular virus, for example, rabies virus, for example, adeno-associated virus, for example, herpes simplex virus. So we can use those viruses as a cargo or as a truck, and we can package some green fluorescent protein or active channels or protein of interest. This has led to the growth in the field of genetic engineering to target a specific brain region, not only specific brain region, but with the introduction of transgenic mice, what you can do is you can target your protein of interest, whether it's a fluorescent protein to map neural circuit, whether it's to test functionality of a neural circuit, like an active channel, which can lead to, for example, sodium entry of sodium into the cell and lead to persistent activity of the cell. So, I mean, with the transgenic mice now, it has been feasible to target a specific cell type in any brain region, even if there are like hundreds of cell types in one brain region. It's a very complex nature of things operating in brain. So brain is just not as simple as a liver, for example, composed of few different cell types. It's probably a permutation and combination of several different phenotypes. Even some cells express some of those neurotransmitters together. And they release it concurrently, or they release it differently to organize different functions. For example, dynorphin and orexin are known to be co-expressed together and co-released together. Glutamate and dopamine, glutamate and GABA, for example, or GABA and dopamine. So the neurons have capability to express number of neuropeptides and neurotransmitters and select what they're gonna release at the postsynaptic cleft in order to regulate neurotransmission. So the idea is there has been lots of advances in transgenic mice, our ability to target specific cell type. There are advances in our ability to trace these neural circuits. So taking advantage of such recent advances in technology, we did a retrograde chemical fluorogold tracer deposition in cuneiform region, and then in a TH iris Cree, this is TH pertains to tyrosine hydroxylase. So a Cree is is an enzyme, recombinase enzyme. So it acts like a lock and key with a flux constructs. So what this animal has is all the brain cells which have tyrosine hydroxylase or messenger RNA for tyrosine hydroxylase, you can tag those cells to express another enzyme, and this enzyme will only act on the fluxed counterpart in the virus. So this is the AAV refers to adeno-associated virus, and this is a human synapsin promoter, which is fluxed, and it also expresses a fluorescent protein called enhanced yellow fluorescent protein. So when we deposit this in those A13 cells, which I showed you a moment ago, we expect it to send projections to everywhere in the brain where it sends projection, including the region we found it sends projection to using a rather very primitive approach of using a chemical tracer. So this is another recent advance which has enabled neuroscientists to explore these connections with a tremendous ability. So I mean, historically, you must be aware with the or well aware how you can cut tissue sections and you can do histology or immunohistochemistry, but you are going like this is not high throughput. You cannot observe everything at once, and particularly structures like brain, they are not transparent, would not allow you to look and see what's inside. So with this development of a new technique called tissue clearing, you can clear any tissue, or you can clear the entire organism. What it does, it takes away most of the lipid, as brain is highly composed of lipids, and then it's as clear that you can see through it. So as you see here, the lines or the grid lines are not visible when the brain is placed on a grid line on a slide, where once you clear the same brain, you can see through it. So this is called clarity, or it has different names, but essentially this is tissue clearing, making the tissue transparent. So as you see, and then coupled with the light sheet microscopy, we have recent advances to image the full brain in like nearly less than 2 hours. So which is tremendous. You can visually visualize. You can go now into the brain and see all the cells in red, for example, are expressing tyrosine hydroxylase. You can see the circuits where these cells are projecting to. So this is now possible to start making a connectome or a map of these neural circuits, but this does create a problem in terms of huge amount of data. Like you need like several hundred terabytes of space in order to do such computations of neural circuits revealed from this powerful technique of connectomics. So once you know that, oh yeah, so this is spread of all the dopamine neurons in the brain and where they're connected. So similarly, I mean, coming back to the point, we have seen that the cells in A13, which express fluorogold, which was coming back from the A30, the cuneiform, and also which express TH in the red, which is rate limiting enzyme for dopamine. And they also express YFP because we injected those cells with enhanced yellow fluorescent protein. And now, once we went back to look at the in a high resolution section in midbrain, we indeed saw several axons and axon terminals shown here in green is YFP, and the same length of axon has synaptic boutons or terminals labeled in TH as well, which is rate limiting enzyme for dopamine. So this is very encouraging that indeed it's a synaptic connection, but we still don't know what are the postsynaptic connection. So we further confirmed the nature of this synaptic connection using another virus. So this is like a fancy toolbox for a neuroscientist. Now you can construct any given number of arrangements of proteins, fluorescent proteins or channels to target neural circuits. So what we have done in this particular custom virus is we have labeled the axons with green fluorescent proteins and the terminals with mRuby, which will lead to red color. And again, we saw that A13 neurons, which express TH, which are dopaminergic, they do send projections all the way to the cuneiform and other brain regions. And you see terminals, presynaptic terminals in this case, are labeled in mRuby. So this all gives confirmation that there is a microcircuit. So this is just one microcircuit. There are millions of microcircuits in the brain, and this is possibly why, I'll come back to this point later, why the traditional pharmacology has side effects. For example, the obvious side effect of taking a weight loss medication has been reported to induce suicidal thoughts. Why does that occur? So I mean, that actually lies in the understanding how the neural circuit which regulate metabolism in the brain, they also regulate emotional behaviors. So I'll come back to that point later. And this is why probably to treat brain disorders, probably drug targets probably won't work as efficiently as they work in peripheral organs. So in a sense, I will wrap it up. There is a microcircuit from this A13, a novel region in hypothalamic region, which express dopamine. So classical flow diagram is depicted in the black. So this is a very basic concept how locomotion is induced via brain. So basal ganglia, of course, from the cortical inputs send projections to the midbrain, and this midbrain cells act as an on and off switch. But there is another on and off switch in caudal brainstem, and then this is routed to the spinal locomotor networks or CPGs, and then we see activation of muscles and followed by locomotion. But what we have seen, and other people have started to show, that the substantia nigra region, for a classical region known to project to basal ganglia, is also sends a parallel command to mesencephalic locomotor region. So what we have started to see, our A13 region also sends a parallel command to mesencephalic locomotor region. So the redundancy, so this goes back to the phenomena that neural circuits are redundant. So there must be, and there are redundant neural circuits to organize and execute similar behavior. So, so now we need to test the functionality of this circuit and how this is involved in Parkinson disease etiology, where the SNC is gone, and you cannot evoke locomotion, and you see freezing of gait and tremor in Parkinson disease. So we have collaborated with a neurosurgeon here who does a human patient deep brain stimulation in various brain regions. The idea is to find new neural circuits and then use the deep brain stimulation or another brain stimulation paradigms in human patients, whether it can be a source of alternative source of dopamine to this region, and then we can restore locomotion. This project is currently ongoing, but we'll keep following this question because the basic science actually has n number of questions in order to understand the circuit, the nature of circuit and the function of circuit. So we want to know what is the function of this A13 MLR circuit we discovered, and how it activates specific, like how would we activate specifically A13 neurons without activating any other neighboring cells, you know, and how to specifically activate A13 neurons which are only connected to this MLR. So these kind of technical challenges we have been able to overcome using new technologies. And this is another fascinating technique. This is also called DREAD. So the word DREAD means designer receptors, which are exclusively activated by designer drugs. So the image on the left, you see, you must be aware with the G protein signaling in the cell. So this, there are different types of channels and in this plasma membrane of a cell, and GPCR is one type of channels and which respond to an endogenous neurotransmitter, for example, to acetylcholine, for example. There are several classes of GPCRs. And this opens the channel, and then you see that the ions rush in. There is flow of ions, and then the neurons are excited or inhibited. So now, with the protein engineering, it has been able to, you know, incorporate new channels in any given cell. For example, I could go and target my A13 dopamine neurons specifically in a given brain region, only those cells, and incorporate this protein pumps or channels, and then which won't be you know, responding to endogenous neurotransmitters, but will only be responding to artificial synthetic ligand, which is a designer drug. And the beauty is the drugs can cross blood-brain barrier, and you can do it systemically. So currently, this DREAD has been probably in some cases has been permitted to be used as a viable option probably for clinical trials. So you can read more about G protein signaling and chemogenetics. So how we will test the function of our given circuit. So as I said, there is A13 neurons which are dopaminergic. We use a transgenic mouse again, where Cre recombinase can be tagged to these cells which express tyrosine hydroxylase, and then we'll target two different viruses, either the virus which will carry an active channel or GPCR, which is HM3DQ. So as you know, I mean, you must have read as, GQ, GI, like in terms of different growth phases of the cell. So the GQ activates the cell, whereas GI inhibits the cell activity. For example, in this case, we try to activate the cell, so hence we use HM3DQ, which is a GQ protein, and then it does a second messenger signaling using calcium, and it activates the cell. And or you can use a control virus, a mock virus, which does not carry the channel, but it only carries the fluorescent protein, which will not result in any activation of the actual neural circuit, but just rather tracing, and then there won't be any behavior. So this is a control group. So we habituated these animals, and you can either use a vehicle or an actual artificial ligand, which is CNO, which is a clozapine and oxide in this case. And you can test the mouse an ability to move in an open field, and you can trace that. So this is just its example trace. So first of all, it's imperative to see our vehicle and control mice, which injected with just the tracer do not have any obvious behavioral effects to the artificial ligand. And we have shown both black and then blue that when the animals are given a vehicle or the artificial ligand, we don't see any different effect post-injection in terms of their locomotor activity. Whereas pay attention to the animal on the top and the animal on the bottom, they're both moving normally in and around the open field before we give the drug. And as soon as we give the intraperitoneal injection of drug, the animal on the top, which has an active channel, starts to frenetically move around the cage, and it keeps moving on until the drug effect lasts. The problem with this is typically the effect of drug lasts around 4 hours. So this poses a challenge in terms of, for example, this CNO can also be taken up by the terminals. And as you know, these neurons are going towards several different brain regions, including our region of interest. So it's hard to contain a microcircuit, or it's hard to study the effect of a particular microcircuit. Yes, we can say the activation of all cells in A13, which are dopaminergic, leads to this excessive locomotor or hyperlocomotion. But it's not sure whether it's mediated with the mesencephalic locomotor region. To do that, we need a more spatial and temporal control over the activation, such as switch on and a switch off. So ability to just switch on for a millisecond or even sub-second level, and then just quickly turn it off, and then do it with a circuit-specific manner. And picture on the right, you see this blue-green algae. And this is very fascinating. Like this simple organism has been used to extract a transmembrane protein or a pump, a sodium pump, because these animals have ability to respond to the UV light or light in a certain wavelength. So and then move their flagellas or it introduces movements. So these microorganisms have been fascinating tool for neuroscientists to extract such fascinating proteins and channels from them. And then what you can do using similar transgenic mice, you can now extract a sodium pump from here, and you can target to the brain cells you want to test function for example, and then shine the blue light on that, and then you'll see the animals will respond accordingly. Then now you can test the behavior of these neural circuits, like what kind of behaviors are governed by which neural circuits. So what we did, so this protein, which is called channel rhodopsin, is a sodium pump. So we package it to adeno-associated virus, which will specifically go and target A13 dopaminergic cells because we are using a transgenic mouse. And then we inserted optic fiber in the brain, specifically above the A13 region. And now we have ability to control at a minute level or even at a millisecond level, you know, giving a pulse of 1 millisecond all the way up to several hundred pulses of like ranging from with a different frequency and amplitude and power. So now we can really go and do electrophysiology and behavior of these cells. And I'll show you. So this video will show you the animals is moving normally before the stimulation comes. And as soon as the stimulation comes, the blue light, we start to shine the blue light right now, and then the animal starts to walk. And it will stop and terminate the locomotion as soon as you turn off the light. So so we have discovered a switch on, switch off responses in the hypothalamic region, whereby we can evoke these locomotion responses. But there are still several questions. I mean, what is the context of locomotion? Why these animals are running around? I mean, are they scared? Is it an avoidance response? Is this an escape response? Or do they like it? Because before you start stimulating brain neurons, you need to know that whether they produce aversive or appetitive effect, because that's the detrimental. If the animal is showing aversive behavior and you see locomotion, I mean, you cannot simply so the next step, I mean, we have a couple of different projects to test this circuit and ability of this circuit to see whether we can use a spinal cord in a spinal cord injury. We can test this ability of this circuit, or we can test the ability of this circuit to improve Parkinson's disease. This is currently ongoing. I won't go into details of those, but let's keep moving on. So I have shown you the zona incerta and a tiny region in zona incerta, which is dopaminergic, projects to cuneiform. And if you activate chemically the cell bodies here or with a blue light here, you tend to see increased locomotion. Still, I haven't shown you whether you can activate a microcircuit. So we have started to do that. So, I mean, this is another example. Some of those cells are also excitatory. They express glutamate. So the calcium calmodulin kinase is also another promoter we use to target excitatory neurons. And we harbor sodium pump channelrhodopsin in those cells. And then instead of stimulating cell bodies, what we do is we go back to the other end of the microcircuit and place the optic fiber at the end at a presynaptic terminal. So now what you can do is, instead of sending signals everywhere in the brain where these cells are connected, you can discretely test the hypothesis that what this microcircuit is doing. So, and then you can look at different postures and different forms of locomotion or movements, which possibly suggest different behavioral manifestation and emotional manifestations. So, like as I said, this is why we need to study exploration and escape neural circuits, and explore. And it's for an animal standpoint of view, it's important as well to keep exploring dynamically changing environment for food, predator, mates, and also scanning the environment for a potential predator. So let's look at this video. Now we have ability to excite a microcircuit, which is excitatory in nature, all the way from A13 to cuneiform. And when the light comes on, you'll see a rapid response, and which is terminated at the end of light. So you see the lights come here on the top left. As soon as the light comes on, the animal freezes for a moment, and then it does a vigorous escape bout of locomotion, which is very rapid. So which is totally different phenotypically from what it was doing earlier. So this is kind of an escape response, which we have seen in the another video, like the high-speed locomotion events such as these are called darting. So the animals do dart on the presentation of a threat stimulus. They freeze for a moment and then they dart. But there is another concept behind this. So the animals do not, so the response of this threat does not persist for too long. I mean, it's not adaptive. So the acute response to this threat is costing energetically, because the animal has to move at a very fast pace, involving its cardiovascular, autonomic outflow, also respiratory needs, also metabolic needs for the muscle, for the whole body metabolism. So I mean, it's energetically costing. Why would, there are two questions. Why, like, the animals cannot afford to escape every time they are presented with the stimulus. So they have to filter out which stimulus are important, and which they have to condition those stimulus. And some of those stimulus, it's not adaptive to condition. For example, as I said, reflex action. Anytime you touch the hot surface, you have to have a knee-jerk reaction, and you have to put your hand away. Otherwise, you'll get burned because it's not adaptive to take away that reflexive nature from the motor action. So, but yet on the other hand, the animal tend to seize, that initial emotional disturbances which were carried with this explosive locomotion and the presentation of that stimulus. The animal must come back in order to feed itself, for example, and that's what you saw in the other video. So the question is, what are the neural substrates involved in termination of this fear response or a threat response for forage yourself? And we have discovered such antagonistic circuit. So what happens, the animal on the left and animal on the right, you'll see the animal on the left has been targeted with the GABAergic cells in zona incerta, or the A13 region, have been targeted to express that channel rhodopsin. The blue light comes in, the animal starts digging, and it discovers a food pellet from the bedding, and it starts to feed itself. So the GABA is inhibitory, leads to inhibitory tone over, and possibly this GABAergic circuit is acting antagonistically to the excitatory circuit we saw. And again, you'll see time and again, and you see the very rapid termination of feeding behavior as soon as the light go away. So the animal starts digging again when the light comes back, and you can do it multiple times. This is fascinating. Even with any length of stimulation, the animal will start digging and will start eating. So these neural circuits are fascinating for some disorders like binge eating, for example, which is prevalent and leading to chronic obesity, for example, or diabetes or metabolic syndromes. Why people cannot, even drinking, for example, even increased drug seeking and increased drug consumption. So these new neural circuits, maybe this is like probably an overconsumption, where, because as I explained, threat and safety are a mixed stimuli present in the environment. In high or extreme threat, the animal execute defensive response, whereas in high safety, the animal execute normal physiological functions such as feeding, foraging. But this seems like there are circuits in response to acute threat or a chronic threat, or a chronic or acute stress, for example, in post-traumatic stress disorder, which can increase drug consumption. Similarly, stress has been shown to increase food seeking, for example, and food consumption, particularly palatable high-fat diet. So, I mean, stumbling upon these kind of neural circuits would help us to understand how these different emotional behaviors are regulated, as well as which evoke different kind of movements not only movement, but also maybe overlapping circuits for doing several different behaviors with a similar redundancy of neural circuits. So we have started to put out a huge connectome. You see a pictogram on the right, different forms of locomotion done with different types of cells. When you excite those in green, or when you inhibit those with in red, the animals execute antagonistic movements, generally, largely you see. And sometimes this effect is heterogeneous. For example, excitation may not always be activating the neural circuits for escape. Sometimes it leads to freezing, for example, which is also part of defensive behavior. And partly my work involves, studying locomotor circuits, but I'm more, because my background, as Dinesh has explained, has been in studying metabolism as well, and studying brain behavior and plasticity. So I'm stumbling on these neural circuits, which might be possibly regulating the different kind of behavioral manifestations of emotional eating, for example. And we do know, and from my previous study, we have been able to show in a study conducted in Montreal University, that brain reward circuit, which is a separate talk, that how this brain reward circuit can leave us vulnerable eating high-fat diets, and especially under stress. And this is a two-way street. Increased stress can increase food consumption, but also increased food consumption and metabolic disorders can increase stress, and depressive and anxiety syndromes and many other phobias. So metabolism, there is a multitude of behaviors done simultaneously, which involve not only motor function, but autonomic, metabolic, and stress-related behaviors, emotional disturbances. Studying those circuits one by one would create a larger connectome and a deep understanding of these microcircuits, because current pharmacological drugs and even novel pharmacological drugs may not be able to cure central nervous system disorders because of the widespread and heterogeneous nature of different neurotransmitters, not only different neurotransmitters, but the possibility of redundant circuits. So in summary, what we have studied today was there is a novel and parallel dopaminergic command to the brainstem locomotor region, and that they may have functional implication for spinal cord injury and Parkinson's disease. We are currently testing these possibilities. And then there is a novel excitatory circuit between the zona incerta A13 region and brainstem locomotor region, which is relevant to escape, as we saw. And it could be relevant, for example, acute response is adaptive for survival. But we have been seeing war veterans, for example, even survivors from emotional traumas. They developed panic disorders such as post-traumatic stress disorder, and many other brain diseases involve emotional and motor disturbances, which are reminiscent of increased anxiety, stress, and probably malfunctions in coping strategies. And escape is part of an active coping. So when this active coping goes wrong, and when there is a chronic presentation of the stimuli, the circuit may turn from adaptive to maladaptive nature, and this will ensue pathology. So we are proposing to study the acute and chronic activation of these circuits and how this can ensue maladaptive changes in the neural circuit, and also induce plasticity at the cellular level, but also at a behavioral level. And we also studied today the novel inhibitory circuit from zona incerta to brainstem region, which does an antagonistic role. It initiates exploration and also induce food intake. And as I said, it might be relevant to understand food seeking, excessive drug seeking, or binge eating disorders, for example. Why people do that, so binge eating is one of the eating disorder. Similarly, increased drug consumption or binge alcohol drinking is problematic in youth and other populations. So explaining this circuit and how emotional disturbances from the environment can affect this neural circuit will enhance our understanding to come to or discover new targets. So yeah, this is a picture from Calgary. And this is downtown for the Calgary. And behind, in the backdrop, you see Rocky Mountains, which are always have fresh snow there. And it's like, and I mean, the point here is, I mean, it presents a very nice environment stimuli to go hiking, which is a positive effect. So I'll end my talk here with this note, and I'm ready for any questions you might have. Thank you very much. Yeah, please feel free to type any questions you might have, and then we'll try to discuss the possible questions.
Dr. Dinesh, can you hear me?
Yes, we can hear you.
Hello, Dr. Dinesh. Dr. Dinesh, am I audible?
Yes, I can hear.
Dr. Dinesh, am I audible?
Okay, so yeah, so the question is, can Parkinson's disease and Alzheimer's syndrome affect be controlled by the present finding? We don't know. I mean, this is very complex scenario in order to find cure. You know, like so. As I said, we don't know much about these neuronal circuit and dysfunction involved in Parkinson's disease. I mean, the traditional view is there is a neurodegeneration in dopaminergic neurons in substantial nigra, but there are new studies which are indicating in both cases, Alzheimer and Parkinson, new developments which might explain why these circuits in the first attempt, like why they go maladaptive. So I mean, with we have a current experiment in place to study whether we can recover, the right word is to recover some of the lost functions in Parkinson's disease. The cure is probably is not possible and without a remission until we don't have a deep understanding of how these neural circuits operate under normal conditions and under different environments, and what are the susceptibilities. So it's like, but current deep brain stimulation techniques, there has been tremendous improvement in our ability to use deep brain stimulation and other forms of brain stimulation in human population, in human patients, to discover new circuits and quickly go back and test the ability of these circuits with the brain stimulation techniques to see if we can recover some of the lost functions. I think that's the right word for the brain disorders. I mean, cure is probably, like, as you know, some of the disorders, it's only possible to cure some of the lost functions. So there is another question from Dr. Kritika. How are the neuronal circuits dysfunction restored in beta amyloid and plaque independent disruption in Alzheimer's disease? This is at the animal model level, there are several studies explaining possible beneficial effects of antioxidants ranging from different types of compounds, I don't know much about Alzheimer's disease, and currently on top of my list are diseases which have a clear motor manifestation, and such as Parkinson's disease. That's what we are taking currently. There is a huge improvement, but we still are discovering and understanding what is Alzheimer's disease exactly. Initially it was thought the amyloid plaque was the culprit, but new studies are indicating there is probably it requires change in our understanding with the new discoveries, with new tools, and we have more data coming up to suggest what are the subtle changes in the neuronal cells and circuits which might be responsible for these things. And as you know, if you're aware with the beta amyloid plaques, these are not the culprits. This is an acute adaptive response in the cell which goes awry in Alzheimer's disease. So I will leave that there. And does the one that work in animal model work for in human? How to proceed? Very challenging issue. The mouse is not a man. So that's to put it like that. If you really have to criticize why we should study even mouse model. But in beginning of my talk, I explained, I could give some explanation in favor of my choosing the animal models, it is a mammal and a fairly simple mammal. It has mostly display homology to genotype or gene pool to very close. And but still in many cases, particularly in pharmacological cases, when we don't see some things which work in mice and rats don't work in human beings, possibly I think there is another culprit to blame. Not proper testing for neural circuits and behavior. For example, the example I gave, weight loss drugs. There is a commercial rush or a business problem here. The drug companies have to come up with a new drug in a quick time span, but the drug efficiency and the requirement for testing currently involve a very basic screening or batteries without the proper understanding how these things are affecting neural circuits at a circuit level. So the key priority is safety, efficacy, but even a drug is safe and and high efficacy, we have seen it often leads to some side effects, or all drugs have side effects. And particularly for the drugs which are targeted at central nervous system, often have side effects. Like weight loss drug, people complain about suicidal thoughts. And there is an answer to that. I mean, if you know about neural circuits and brain reward circuits, you will know these are the similar circuits which will push you to eat a dessert after a meal. And these are the same circuit which will be affected in depression and anxiety. So, I mean, targeting a circuit with a blunt force like a hammer would not help in treatments. So that's what we are probably debating here. The understanding is important for neural circuits. Whether the mouse behavior translates exactly into human beings would be can be debated at another level. But first, no doubt, we need more understanding. So is artificial neuron culture possible? I mean, yeah, people do it all the time, artificial neuron cultures, and you can read more about it. And if you need more information, I can send you more link. Yeah, and yeah. So let me know if I have missed any question.
Yes, over to Dr. Dinesh, please.
Yes, sir. Am I audible?
Yeah, sure.
Okay. Thank you, Dr. Sandeep, for such an informative session. I would be sharing Dr. Sandeep's number, email address to all the participants so that they can contact him later on if they have any more information to acquire. So now I would like to invite head of the department, Professor Puneet Puri, for extending a vote of thanks to the speaker as well as to all the audience. Professor Puneet Puri, please.
Thank you, Dr. Dinesh Lakhanpal. Dr. Sandeep, I'm really honored to listen, and I also thank all the August Assembly here who have spared their precious time to hear from such an wonderful lecture from your side. It was really mesmerizing, and the knowledge which was there in your lecture is really good. And I had seen the presentation. Overall, there was some electricity problem going on in India, you know very well. But I also invite you to India. Whenever you come, you please visit our department, PG Department of Zoology. We are really honored to listen from you all. And thank you for such a good interactive session. And I will be failing in my duty if I would not thank Dr. Dinesh for arranging such a wonderful lecture and a wonderful speaker like Dr. Sandeep. Thank you very much, sir. And you are most invited to visit our PG Department of Zoology, DAV College, Jalandhar. Thank you very much, everyone. Thank you, Dr. Dinesh.
Thank you, sir. Thank you. Again, I have one very important announcement to make here. I have already shared the link for your feedbacks in the chat. So you are requested to please fill it up as soon as possible because we would be accepting the feedback for next half an hour only. Only those people who are able to fill up the feedback would be given the certificate. So please take it as most serious. Take it seriously, and so I believe that we now must leave the meeting. So please complete these feedback forms. Thank you everyone. Thank you for participating. We would be organizing more such events in the coming time if COVID allows us. Thank you everyone.